And this is also telling you that it is from the exon of the middle of the coding region. So remember that exons are expressed as proteins. It's in the middle of the coding region, and it wants us to determine the mRNA that's transcribed from it. So in order to do this, we're going to have to find an open reading frame. Now, because the codon language is a triplet language, right? Each codon has 3 nucleotides. There are 3 possible beginning points for each strand that we can look for an open reading frame. Now remember that open reading frames are ones that do not have a stop codon within them. So what we're going to look for are the stop codons. Now in RNA language, that's UAA, UAG, and UGA. However, in DNA language, we're going to change the U's to T's. So we're looking either for TAA, TAG, or TGA. Because open reading frames are found in the coding strand of DNA, and therefore the mRNA, we're going to look from 5' to 3' because that's the direction that mRNA is read. So looking at the top strand first, we're going to start at the 5' end, and remember there's 3 possible positions that we can start looking for open reading frames. So I'm going to do each one and erase, and then we'll keep going. So for the first one, if we start at this first nucleotide, remember we're reading from right to left here, and we're going to keep an eye out for these stop codons. So this first one is not a stop, neither is that, but then this one, if you're reading it this way, you see TAA, which is a stop codon in DNA language. So this first possible reading frame is not an open reading frame. So now let me erase this. And let's start from the second nucleotide. So right away we see TAA is a stop codon, so the second reading frame is not open either. Now let's start from the third. We have AAG, that's fine. TTA, fine. ACT, okay. TTC, GAC, but now this one you see TAG, which is a stop codon, so this reading frame is not open either. So now we're going to look on the bottom strand, the complementary strand, and see if there's an open reading frame there. Remember, we're going from 5' to 3', so we have 3 possible frames, 1, 2, or 3. And let's start with the first nucleotide. AAC, CTG, GAC, and then you see TAG, which is a stop codon, so this first frame is not open. Now with the second nucleotide, we see CCC, TGG, ATC, AGT, and so on. Remember to keep an eye out for those 3 stop codons. CT and TAC, that's not stop, so it looks like this second frame on this complementary strand is open. And let's just double check that the third frame, whether it's open or closed. Okay, so starting with the third nucleotide now, CCT, GGA, CTA, GTC, GAA, AGT, and then you see TAA, which is a stop codon, so that's good that this third frame is in an open reading frame. So the open reading frame is in the bottom strand, and we'll write 2, that starts at nucleotide 2. So the problem is asking us, determine the nucleotide sequence of the mRNA that's transcribed from this region. Well, remember that open reading frames are found in the coding strand. And coding strands are analogous to the mRNA, except that the T's from the DNA are changed to U's in the mRNA. So the mRNA sequence here, we're just going to copy the coding strand, which is this bottom strand, and let's start at the second nucleotide because that's where the open reading frame begins. So the mRNA is going to be 5', CCC. Now let's change the T's, remember, to U's. And then you just continue along and copy every other nucleotide except the T's are going to be used. So I'm just going to write that, and you can follow along and see if you got the same answer. Okay, so remember, it's going to go in the same polarity as the coding strand. So let me just label that so you can see this later. This is the coding strand, and this would be the template strand. Okay, so this is the answer for part B. They wanted the sequence of the mRNA transcribed from this coding region. Now part C says to determine the primary structure for this part of the protein. Now usually when you're determining primary structure, which remember, primary structure equals the sequence of amino acids. When you're looking for the sequence of amino acids, usually in a protein, you're going to look for the start codon to say that this is where it starts. However, from part B, they told you that this sequence that they gave you was from the middle of a coding region. So we're not going to look for a start codon because that happened upstream of it. So we're just going to start with this first nucleotide because that's where the open reading frame started, and we're going to see which amino acids are encoded for. So if you don't have a genetic chart out, you might as well, you should take it out now. And we're looking, starting here. So the first codon is CCC, and that encodes for proline. Then you have UGG, and that encodes for tryptophan. ACU encodes for threonine. AGU encodes for serine. CGA is arginine. AAG is lysine. UUA is leucine. ACU is again threonine. And finally, UAC encodes for tyrosine. So you might want to write the polarity of proteins, and proteins run the direction from the N terminal, the amino terminal, to the C or carboxy terminal. So let's just add that here. You can write N, or you can write NH2. And then here you write C, or you can write COOH. So this is a polypeptide from the middle of the sequence. And because it actually is in the middle, what we want to write is N dot 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 to show that there are amino acids before this, and then tyrosine dot 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 C to show that there are amino acids after this. So from the middle of this region, this polypeptide is proline, tryptophan, threonine, serine, arginine, lysine, leucine, threonine, and tyrosine. Problem 3. A complete eukaryotic gene for a small polypeptide was sequenced, and the results are shown below. The entire sequence on the template strand is transcribed into RNA and produces the protein methionine, protein, tyrosine, arginine, and aspartic acid. Part A. Determine which strand is the template strand and in which direction RNA polymerase moves along the template as it transcribes. So the first way to find out which is the template and which is the coding is to find out what is the possible mRNA that can give that polypeptide. So we're going to use the genetic code chart to do that. So we'll write mRNA. Now the first amino acid is methionine, which is the start codon and is AUG. Remember that mRNAs are read from 5' to 3'. So we know the first is going to be AUG. Then proline can be made, let's see, there's 4 here. Let me just make sure there's no other prolines. Okay, it looks like those are just 4. So we can write CC dash because the third can be any of the 4 nucleotides and it will still encode for proline. So we'll write CC dash. Then we have tyrosine, and tyrosine can be UAU or UAC. So I will write the C down there. Then you have arginine, and arginine can be these 2 or these 4. So it could either be CG dash or it can be AGA or AGG. And then you have aspartate or aspartic acid. So aspartate here, let me just make sure those are the only 2. Okay, so it looks like those are the only 2 aspartates. So it's either GAU or GAC. And then it ends in 3'. So to find out which is the coding sequence, now remember the coding strand is the one that's analogous to the mRNA sequence, except that the U's from the mRNA are changed to T's in the DNA. And we could see which one is most similar. So if you look from 5' to 3' on the top strand, you see ATG, which in DNA that matches with the AUG. Then you see CCG, and this G can definitely be from this dash. Then you have TAC, and when you change T to U, that could definitely be this amino acid sequence codon sequence. Then you have AGG, which matches down here. And then the next codon you see is GCC, and that doesn't match. So the next codon that we're looking for from the mRNA is either GAU or GAC. And you can see that if you go down here, you see GAC. Now the reason why it's okay that we skipped some nucleotides here is because, remember, this is from a eukaryotic gene. Now eukaryotes have a process known as splicing, where a complete DNA gene can be spliced into smaller pieces for the mRNA, and those are the exons that are expressed, right? So this right here could be an intron. So as we can see, this top strand seems to be the coding strand, right? Because it matches up with what our mRNA should be. So let's label this top one as coding, and therefore this bottom one is template. And it's asking, which direction does RNA polymerase move along the template as it transcribes? Remember that RNA polymerase moves 3' to 5' on the template strand to make a 5' to 3' mRNA. So RNA polymerase is going to go from left to right. It's going to go this way, from 3' to 5'. So that answers A. And then for part B, it says determine the sequence of the processed mRNA for this gene. Now processed means after splicing, right? So let's extend the page a bit. And now we're going to write mRNA again and just clean it up a bit. So we know, again, this first codon is going to be AUG. Then the second one, we know it was CC dash, but remember we said that it matched with this G up here, so we know now it is CCG. Then you have TAC, so in RNA it's going to be UAC, because that matched with the coding strand of DNA. Then the next codon matched with AGG. And then finally, the next codon after the intron is spliced out is GAC. And it didn't tell us anything further, so this could also be removed through splicing. So this is the mature mRNA sequence that you get after this eukaryotic gene is transcribed and then gone through splicing. Now let's do the true and false section. Problem 1. The central dogma of molecular biology can be simplified as DNA to RNA to protein. Now this is true, as you might remember from the theory videos. So DNA is transcribed into RNA, and then RNA is translated into protein. 2. Similar to DNA polymerase, RNA polymerase needs a short RNA primer in order to begin synthesizing mRNA. Well, this is false, and that's because RNA polymerase does not need a primer. You remember that DNA polymerase does need that short RNA primer, but RNA polymerase can just start synthesizing without any primer. So we could say false, RNA pol does not need a primer. In eukaryotes, RNA polymerase II is responsible for transcribing tRNAs, the 5S rRNA subunit, and most snRNAs. Well, this is something you probably have to go back into the theory to find the answer to, but this is actually false. So in order to make this right, you can change it to pol RNA polymerase III. So that is the one that actually transcribes tRNAs, the 5S rRNA subunit, and most snRNAs. But polymerase I makes rRNAs. Polymerase II makes mRNAs. And then again, polymerase III is what transcribes what is said in the question. Question 4. All mRNAs have a coding region that sits between two untranslated regions. This is actually supposed to say sits. So that is true. An mRNA, messenger RNA, has 5' and then it has the 5' untranslated region. Then it has the coding region, then a 3' untranslated region, and then the 3' terminal. 5. Poly-A tailing in the RNA modification is best described as removal of intervening sequences. Well, this is false. The removal of intervening sequences is known as splicing. The poly-A tailing in the RNA modification is where polyadenylenes are added to the end of an RNA. 6. Crick and colleagues used frameshift mutations to verify that a codon consists of 3 nucleotides. And that is true. They saw that if they frameshifted over 1, they would get a different codon, different amino acid being encoded for. If they shifted over 2, they would also see it, but if they shifted over 3, then you would just get the next amino acid. So they verified that the codon is 3 nucleotides. 7. The last 2 letters in the codon seem to be more important than the first in determining the correct amino acid. So this is talking about wobble theory, right? And we're going to assume that when they say the codon, they mean from 5' to 3'. Now if they do that, that actually is false. And I'll write the explanation down here. When you have 5' and then 3 spaces, right? Because there's 3 nucleotides in each codon. These first 2 are actually more important than the last. So this last one is the one that's not as important for base pairing. And therefore it can still base pair with an anticodon for the correct amino acid if these first 2 are correct. 8. The A and P site are located within the small ribosomal subunit. Now this is false, and it's just another fact you have to know, that the A and P site are located within the large ribosomal subunit. Okay, so if you did well on this, that's great. If not, you might want to go back to the theory and rewatch it or just reread the notes. But this concludes the review for protein synthesis and RNA transcription.